Good morning, students. How are you all? Today I am going to discuss about the structure of chromosome. Okay, we are discussing the lesson regarding the genetics. Am I right? Heredity. So in heredity, chromosome is a important part. Clear? What is chromosome? What is the parts of the chromosome? How these chromosomes involved in replication process? I mean making the protein synthesis. Okay, clear? So you know what is chromosome. Initially, when we see the cell, at the center of the cell, that is which is called the nucleus. Okay, when we look at the nucleus, you will identify the smaller globular structure that is called the nucleolus. Okay, then we will identify the thread-like structure coiled around the nucleolus. Am I right? Clear? What is this thread-like structure? This thread-like structure is called chromatin. This chromatin consists of DNA, RNA, histone and non-histone protein. The chromatin consists of DNA, RNA, histone and non-histone proteins. Okay, around this DNA, okay, there are 8 proteins are identified. Okay, so or else we can say around the 8 histone proteins, the DNA, I mean the chromatins are coiled around. Chromatins are coiled around. So this is, we call it a nucleosome. This is, we call it a nucleosome. Okay. So what is chromatin? A thread-like structure identified in the nucleus. Okay. So when the cell undergoes cell division, அதாவது ஒரு செல் 2 செல்லா டிவைட் ஆகுது, 2 4ஆ டிவைட் ஆகுது, 4 8 16 32 அப்படி டிவைட் ஆகும். when the cell undergoes the cell division, at the time the thread-like structure, the chromatin are condensed and forms chromosome. இப்போ புரியும்னு நினைக்கிறேன். chromatin என்றது அந்த யூசுவலாக அந்த செல்லுக்கு அந்த நியூக்ளியஸில் இருக்கும். ஓகே, இது வந்து த்ரெட்-லைக் ஸ்ட்ரக்சர் போல் இருக்கும். இந்த த்ரெட்-லைக் ஸ்ட்ரக்சர் என்ன பண்ணால் 8 ப்ரோட்டீன்ஸ் வந்து காயில் அரவுண்ட் ஒரு பால் எடுத்து அதை சுற்றி அந்த த்ரெட்டு டைப் பண்ணால் எப்படி இருக்கும், அந்த காயில் பண்ணால் எப்படி இருக்கும், அதே போல் தான் அந்த 8 ப்ரோட்டீன்ஸை சுற்றி இந்த நியூக்ளியஸ் இந்த chromatin வந்து காயில் பண்ணும். ஓகே, இந்த ஸ்ட்ரக்சரை நம்ம நியூக்ளியோசோம். நியூக்ளியோசோம் இஸ் மேட் ஆஃப் the DNA coiled around the 8 histone proteins. Okay, that's we call it a nucleosome. So the chromatin cell division அப்ப condense ஆகும், அப்பதான் visible ஆகும் நமக்கு. Till that it is not visible. The chromosome will not be visible until the cell undergoes the cell division. Cell division அப்ப during the cell divisions, then this chromatin is condensed and forms a chromosome. Okay, this chromosome will be having the genetic material called DNA, because I already told the chromosome is the vehicular transmission of genetic material. So this chromosome carry the DNA. Okay, a small segment of DNA is known as a gene. ஒரு சின்ன பகுதி தான் நம்ம ஜீன் என்றோம். This gene is responsible for the particular protein synthesis. Do you understand what is chromatin? Clear? Then what is chromosome? Then what is the DNA, gene and protein? Okay, and this chromosome, the word, who has given the term chromosome? Who has given this term chromosome? Okay, so Valtier, he has given the term chromosome. So previously, before him, Strasburger, so he is the person who identified the chromosome in the eukaryotic cell. Okay, let's move on. So the term chromosome is given by Algier. The chromosome is having two arms. Okay, that's we call it. It's made up of two arms. These two arms are known as chromatids. These two arms are known as chromatids. These two arms are known as, this is chromatid, this is also a chromatid. Okay, do you understand? The chromosome is made up of two arms. That is a chromatid. So it's each chromosome having a pair of chromatids. Okay, chromosome always exists in the pair conditions. Clear? If the chromatids are similar in shape and size, the randomly similar shape size they are sister chromatids. They are sister chromatids. If the chromatids, the pair of chromatids are similar in shape and size, they are known as sister chromatids. In the random number is a sister chromatids. Sister chromatid. Sister chromatids. If it is different, then we call it a non-sister chromatids. In the random non-sister chromatids. Non-sister chromatids. Okay, clear? Sister chromatids and non-sister chromatids. Sister chromatids both are similar in shape and size and structures. Both, that is we call it as sister chromatids. Okay. So these chromatids are connected in a center point, that is we call it a centromere. This centromere has the grooves, that is we call it a primary constrictions. So that chromosome will be having the grooves, that is we call it in constrictions, and it is having the primary constriction as well as secondary constriction. Primary constriction and secondary constriction. This at the primary constrictions, there is a center point, okay, which connect both chromatids. There is a center point which connects both chromatids, that is known as centromere. Okay, a centromere, a disc-shaped structure. Around the centromere, proteins are there. That protein we call it a kinetochore. Okay, so around the protein, this kinetochore helps to bind with the spindle fibers during the cell division. So the spindle fibers bind down. Do you understand what I am saying? Bind with the spindle fibers. Next thing, at the end, then you are having the secondary constriction. The secondary constrictions. Okay, at the base of the secondary constriction, we will find a round shaped structure, that is we call it an satellite chromosome. Satellite chromosome. This is we call it an SAT chromosome. SAT chromosome. Okay, this satellite chromosome. Okay, fine. This secondary constrictions involved in the nucleolar organizer. So that this secondary constriction has the nucleolar organizer. Okay, nucleolar organizer system. So thereby, and this is involved with the nucleolar organizations in order to produce the ribosomal RNA. Since it is involved in the synthesis of ribosomal RNA, then we call it an nucleolar organization regions. Since the secondary constriction involved in the synthesis of ribosomal RNA, so thereby it is termed as nucleolar organizers regions. Got it? Okay, fine. So here at the end of the chromatids, there is a portion, the small portions called the telomere. Okay, the end. Telo means end. So at the end of that chromatins, there is a small sequencer. Repeated short sequences are identified that forms a telomere. This telomere will have the repeated short sequences 5' to 3', the tag sequences TTA, GGG. Okay, and these telomeres are identified in the Neurospora crassa and human being. Neurospora crassa and human beings. What is the function of the telomere? This telomere prevent unnecessary fusions with the chromatids of the other chromosomes. Okay, next things, and it is involved in the aging, decide the lifespan of the chromosome. Decide the lifespan of chromosome.